So today we're going to be talking about villi and dietary fiber. So this is a diagram of the epithelial lining of a small intestine, and as you can see, it's highly folded into these finger-like projections, which are called villi. And villi basically function to increase the surface area available for material absorption. Let's quickly just go over exactly what materials are absorbed by the villi in the small intestine. So firstly, we have glucose. This is created when starch is broken down into maltose by the enzyme amylase. Just a reminder, starch is a polysaccharide, meaning that it is a polymer of sugars, whereas maltose is a disaccharide and is two glucose molecules attached together by a glycosidic bond. When this glycosidic bond is broken, catalyzed by the enzyme maltase, remember the suffix is what we use to signify that something is an enzyme, then this will form glucose. The second material that is absorbed is fatty acids and glycerol. So lipids are emulsified, meaning that they're broken down into smaller pieces by bile. And this basically means that they have a larger surface area to volume ratio, which means that it's easier for the lipase to attach onto them and digest them into the fatty acids and the glycerol. So then proteins are broken down into amino acids, and in the stomach, this is done by pepsin, which only works in acidic conditions. Then in the small intestine, this is done by endopeptidase. And all of these enzymes are released by the pancreas. So the villi have lots of structural features and adaptations to facilitate the absorption of these digestive products. So firstly, there's the microvilli, and these are basically just even further projections of the epithelial membrane in order to increase the surface area even more. Then we have a rich blood supply, and this basically is just a dense capillary network which allows for easy absorption of the nutrients because they can just diffuse into the bloodstream from the lumen, which is just the space, the hole in the small intestine in which the food passes through. Then there's a single layer epithelium, and this basically means that the diffusion distance between the lumen and the blood is really small. So the nutrients basically don't have to travel very far in order to be absorbed. Then we have the lacteal, which absorbs lipids from the intestine into the lymphatic system. Then we have intestinal glands, or exocrine pits, which are called crypts of Lieberkühn, and these release digestive juices. So these epithelial cells also contain lots of membrane proteins to facilitate the various different types of transport of the digestive materials into the bloodstream. They also have lots of mitochondria, which respire aerobically to produce ATP energy for active transport of nutrients into the bloodstream. I'll get into this in a little bit more detail in a minute. They also have tight junctions, which ensure one-way movement. Basically, they make sure that the nutrients from the bloodstream don't move back into the lumen of the small intestine, because this is counterproductive, right? Obviously, we want to absorb the nutrients into the bloodstream, not vice versa. So the membrane transport mechanisms, which we have to know, there's 5 main ones. Let me just come down here so I can show you. Firstly, there's co-transport proteins. And this is basically when a molecule is transported via the passive movement of another, or via the active movement of another. So for example, we have a co-transport protein which pumps Na plus across the epithelial membrane into the bloodstream. And glucose and amino acids basically attach onto this sodium ion, which allows them to be pumped along with the sodium ion into the bloodstream. Then secondly, we have facilitated diffusion. And this is when channel proteins help hydrophilic food molecules pass through the hydrophobic portion of the plasma membrane. Because if you remember, the plasma membrane structure has these hydrophilic heads, but then the tails are hydrophobic, which means that anything that's polar can't move past them because there's repulsion. That's why we need these channel proteins, which are basically embedded in the membrane and have a hydrophilic center or a hydrophilic passage, which allows for this movement of any polar molecules. So examples of molecules that are transported through this is like monosaccharides, not necessarily glucose, but like fructose, vitamins, and some minerals as well. Then thirdly, we have osmosis. And this is basically when water molecules will diffuse across the membrane in response to the movement of ions and hydrophilic monomers or solutes. So basically, when the solute concentration is higher in the bloodstream, this means that the water potential is also lower, meaning that water will move from an area of high water potential to low water potential through the passive, partially permeable membrane. Then we have simple diffusion. And this is basically for hydrophobic materials, as these can freely pass through the hydrophobic portion of the plasma membrane. So basically, they don't even need this protein. They can just move straight through this. And then once absorbed, these lipids, because most lipids are hydrophobic, will pass into the lacteals, which is this green section, instead of the bloodstream, because they're absorbed by the lymphatic system. Then lastly, we have pinocytotic vesicles. And this is basically the non-specific uptake of fluids and dissolved solutes. So these materials will be ingested via the breaking and reforming of the membrane, and hence are contained within a vesicle. So for example, if we have the membrane here, and we have a liquid which is to be absorbed by pinocytic vesicle, this is basically a form of endocytosis. So the active taking in of bulk materials. This membrane will basically bend and form a vesicle around our ingested material, or our material to be absorbed, which allows it to move into the bloodstream. So the indigestible portion of food, which is basically derived from plants and fungi, so things like cellulose and chitin, is called dietary fiber or roughage. And the reason that we can't digest this is because we don't have the necessary enzymes to break down that plant matter. So for example, if we wanted to digest cellulose, which is found in most plants, or all plants actually, because it's part of the cell wall of the plant cell, we would need the enzyme cellulase, and we don't have that. Certain herbivores do. So for example, cows would have the cellulose, which would allow them to eat grass as their main, or as a really big source of nutrients. So the rate of transit of materials through the large intestine is largely dependent on their fiber content. So roughage provides bulk in the intestine, which helps to keep materials moving through the gut. And it also absorbs water, which keeps bowel movements soft and easy to pass. So there's several health benefits associated with diets that are rich in dietary fiber. Firstly, it reduces the frequency of constipation and lowers the risk of colon and rectal cancer because obviously it essentially helps you to go to the bathroom. It also lowers the blood cholesterol and regulates blood sugar levels by slowing the rate of absorption of nutrients. And it also aids in weight management as it contributes to fewer calories despite consisting of a large volume of ingested material. So if you eat a lot of like leaves, for example, you're eating a lot of cellulose, which doesn't even get digested. So you don't actually intake that many calories. So materials that are not absorbed by the small intestine and large intestine are ultimately ingested from the body as feces. Do not mistake ingestion with excretion because excretion is the removal of a waste product such as ammonia. However, ingestion is just the removal of things that we couldn't digest. It's not necessarily a waste product, it's just not really useful. So a large portion of human feces consists of dietary fiber, basically, which is like cellulose, lignin, chitin, stuff like that. And other things that are present in feces are the remains of intestinal epithelial cells, bile pigments, and any intestinal bacteria that's found in our intestines and stomach. So thank you so much for listening to this video. I hope it helped. Bye.